So thank you and good morning. I want to thank Mark for organizing this symposium and for his kind invitation to present in it. So since my postdoc, I have been very much interested in age-related changes during adulthood. So I mostly focus my effort for the sleep changes occurring between the 20s and the 60s. And even between the 20s and the 60s, you have some very important and significant age-related changes. For instance, if you measure sleep at home with sleep diaries, you already have earlier bedtimes and earlier wake times, and people spend less time in bed. If you measure their sleep with polysomnography, you will notice between the 20s and the 60s, less time asleep, more wakefulness, lower sleep efficiency, less slow wave sleep, and more stage 1 and stage 2 sleep. And actually, those changes are very prominent during the middle years of life. And I will define middle years of life being between 40 and 65 years old during my talk. You also have some changes like shorter REM latency and less REM sleep, although they are less pronounced in the middle years of life and more pronounced later during the aging process. So these are the age-related changes during baseline sleep. However, we also have studied the aging system while it was challenged. And the take-home message for that is that as you are getting older, even starting during your 40s, your sleep will be more vulnerable to many kinds of challenges. For instance, we have shown that if you compare the response of sleep to sleep deprivation in young and middle-aged subjects, middle-aged subjects will have lower rebound of slow-wave sleep after sleep deprivation, showing that they are less able to increase sleep intensity following a sleep loss. Older subjects also show a decreased ability to maintain sleep at an abnormal circadian phase, and this despite sleep deprivation. For instance, in shift workers, after a night shift, usually they spend the entire day before the night working, and then they try to recuperate during daytime at the time that the circadian signal is sending a strong waking signal. We compare the ability of young and middle-aged subjects to be able to sleep and to sleep without interference of wakefulness, and this is much decreased with increasing age starting at the year 40. And we have shown also that the sensitivity to caffeine effects, especially to higher doses of caffeine, is also increased during the middle years of life. So since most of the effects of age during the middle years of life were on non-REM sleep, I got very much focused on non-REM sleep. And I want to present to you two electrophysiological markers of non-REM sleep. We have the sleep spindles, which are bursts of fast wave, like between 11 and 15 hertz. That occurs periodically during stage 2 sleep mostly, and they have a duration of about 500 milliseconds or a little bit higher. And you have those big slow waves also that are higher in amplitude than 75 microvolts and have a frequency of lower than 4 hertz. And of course, they are more prominent in the deep sleep stages. Plenty of animal research and human research have shown that those non-REM sleep oscillations, the spindles and the slow waves, play a critical role in sleep and synaptic homeostasis, in sleep protection against external stimulation, in learning, memory, and cognition. We have found some very important age-related modification in non-REM sleep EEG oscillation. For instance, if you take the slow waves, and here you have young subjects and middle-aged subjects in white, and you can see that you have a major decrease of slow wave density, so the number of slow waves per minute of non-REM sleep. And this is the case both for men and women. You also have changes in characteristic of the slow wave. So the slow wave have lower amplitude, and they show also a lower slope between the positive and negative phase. If you look at the spindles, you can see also some very prominent age-related changes on spindle characteristics. Now I have 3 age groups. So I added in the white triangles an older group, 65 years and older. The middle age are the triangles, the black triangles, and you have the young subjects. The thing that I want you to notice is that you have a decrease in the density, so in the number of spindles per minute on non-REM sleep, you have a decrease in spindle amplitude, and you have a decrease also in their duration. But note that age-related effect is way more prominent between young age and middle age than it is for non-REM sleep oscillation between middle age and older age, like 65 years and older. So most of those age-related effects on non-REM sleep oscillation are occurring during the middle years of life. So we have shown that those non-REM sleep oscillations are also linked with the general cognitive ability of healthy older subjects. So it's more like a trait for the older subjects. So if you take older subjects, the ones that are showing higher spindle density were also showing higher performance on declarative memory tasks, on attention tasks, and on verbal fluency. For slow wave density, it was less, only one variable or cognitive variable was linked to a slow wave density, and that was verbal fluency. We also asked, since spindles are associated to brain plasticity and to cognition, there was a paper that was published in 2014 showing that patients with Parkinson's disease were showing lower spindle density. So through a collaborative work with Dr. Jean-François Gagnon, who was following a cohort of Parkinson's disease patients, for which we had baseline polysomnography in 68 non-demented Parkinson's disease patients and in 40 healthy individuals. And we followed them up for a mean average of 4.5 years. And after 4.5 years, 18 Parkinson's disease patients developed dementia, and 50 remained dementia-free. So the question here was, can sleep 4.5 years earlier can predict the people or the Parkinson's disease patients that will develop dementia after? So we look at polysomnographic sleep, but we were mostly interested in spindle, and actually that was the variable that was significant. So keep with me with this busy slide. I will, oh, you see? So I need to go back. So here you have spindle density, you have spindle amplitude and spindle frequency. And so in the black, you have the Parkinson's disease patients that did develop dementia 4.5 years later. In the ash line, you have the Parkinson's disease patient that did not develop dementia. And in white, you have the control subject. And what you can see for spindle density and for spindle amplitude is that the subjects, the Parkinson's disease patients that did develop dementia 4.5 years later already had reduced spindle density and reduced spindle amplitude before. And actually, these effects were more prominent in the posterior derivation of the brain. For spindle frequency, they also have shown, we also have shown that spindle frequency was lower 4.5 years earlier in the Parkinson's disease patient that did develop dementia as compared to the control subjects, but it didn't differ in terms of, with the Parkinson's subjects that did not develop dementia. So those data suggest in a prospective way that spindles at baseline in Parkinson's disease were associated with the likelihood of developing dementia at follow-up, so 4.5 years later. So my research also is really focused on trying to understand age-related modification in non-REM EEG oscillation to see if they are associated with anatomical and functional changes in the brain. So I am using a multimodal approach using MRI, fMRI EEG, and MEG EEG to see also if those changes could be linked to age-related changes in brain plasticity and cognition. So I will just give you, for a matter of time, the example of two studies that is running in my lab, one that is published, the other one that is not published. And these were talk about anatomical data with MRI, and I will show you also some preliminary data from an fMRI EEG study. So the first study, the question was, are age-related changes in non-REM EEG oscillation associated with cortical gray matter? So cortical thinning is a hallmark of age-related changes in gray matter, and you can see that for some variable of cortical thickness, there is a decrease of cortical thickness across the ages, so between the 20s and the 90s years old. And the previous studies in teenagers have shown that the decrease of slow waves that you also have during adolescence was parallel also to the changes in cortical thickness in adolescents. So we had the exact same kind of hypothesis with the older subjects. So we studied 30 subjects, young and 33 older subjects. All that data that I'm reporting since now, I forgot to mention that, but they are all in the same kind of population. They are very healthy subjects without sleep complaint, without sleep condition, and in very good health. And that was the case also for this study. So all subjects came for a screening night, then we did a polysomnography night and MRI scanning. So the question that we asked was, does cortical thickness explain age-related changes in slow waves? And to answer the question, we use a mediation model. And to use a mediation model, I will have to show you first that in this cohort, and that is not surprising, age was strongly associated with a reduction of frontal slow wave density and with a reduction of their amplitude. To be able to test mediation, I will also have to show you that cortical thickness, after you control for the effects of age, is also associated with frontal slow wave density and amplitude. And I will have to show you that for those brain areas that are showing a relationship with frontal slow wave density and amplitude, that these are also affected by age. And then it will be possible to calculate the indirect effect of age, and then ask the question whether the effects of age on frontal slow wave density and amplitude can be explained by cortical thickness. Oops. So to answer the second relationship, so yes, after you control for the effects of age, cortical thickness is indeed associated with slow wave density and slow wave amplitude. So slow wave density, higher thickness in the inferior parietal, in the middle temporal, in the superior temporal, and in the middle temporal gyrus was associated to more slow wave density, to higher slow wave density. For slow wave amplitude, also higher slow wave amplitude was associated to thicker cortex in the right middle frontal gyrus, in the right superior parietal lobule, in the left cuneus, and in the left lingual gyrus. And these effects are after you control for the effects of age. Now, I told you that I needed to show you that those brain areas were affected by age, and actually most of the cortical regions associated to slow wave showed lower cortical thickness in older individuals, all of them except the lingual gyrus. So up to now, I have shown to you that age, increasing age, is associated with a reduction in slow wave density, and that there were 4 brain areas that were associated after you control for the effects of age with slow wave density and with age. Now the question, we use an integrative model of parallel mediation analysis in order to detect, which of these regions or if all of these regions are explaining the change in slow wave density that we have with age. So we put all those regions into competition in a parallel mediation model, and we found that the middle frontal gyrus and the middle temporal gyrus explain the entire relationship between age and slow wave density. Now, if you do that for slow wave amplitude, so I have shown you that age is associated with a strong reduction in slow wave amplitude, and that there were 3 brain areas that were associated both with slow wave amplitude and with age. And if you put them into competition in a parallel mediation analysis, you don't find here one specific region. It's really the 3 of them together. So it's the global cortical thickness in these 3 regions that explain, but this time not the entire relationship, but part of the relationship, because the residual relationship with age was still significant, meaning that for slow wave amplitude, other factors than cortical thickness are probably at play to explain those changes. So the conclusion is that thinning in a network of cortical regions involved in slow wave generation and propagation, but also in cognitive function, explain the age-related decrease in slow wave density and amplitude. So now the second study, it's about functional connectivity. We were interested to see if age-related changes in non-REM sleep were associated also to changes in how the brain is connected during sleep. And in young subjects, EEG fMRI studies have shown in general, and this is a global result, that there is a decrease in functional connectivity in several cerebral networks during non-REM sleep. And globally, cortical functional connectivity, at least the long-range connectivity between distance areas, decreases with the descent to non-REM 3 sleep. So, of course, since the older subjects have less deep sleep, we make the hypothesis that they will show less of this disconnection across the deepening of sleep. So to do that, we recorded EEG and sleep actually within the fMRI, not for an entire night, but only for 2 hours. a maximum of 2 hours. And we made the data acquisition following a 26-hour sleep deprivation. You can imagine why. I told you that the older sleep is very sensitive to challenges. Believe me, sleeping in an fMRI is a challenge to the sleep. So in order to decrease this challenge, we made subjects sleep deprived. And that was okay for our hypothesis because I told you also that the sleep of older subjects is less able to increase sleep intensity, so age-related changes in non-REM were even more pronounced in this situation. So we studied 16 young subjects and 14 older subjects in those situation. And this slide is showing the sleep. So it's only to show you that, it's difficult with the button. It's only to show you that all subjects slept a sufficient amount of sleep to be able to do functional connectivity analyses. This is showing age-related differences that we found in sleep stages in the scanner, and those differences actually are exactly what you would expect outside the scanner. So the older subjects had more wakefulness, more stage 1 sleep, and way less non-REM 3 sleep, which means that it was not possible to make comparisons with N3 sleep in the older subjects. So then, this is a collaborative work also with Dr. Pierre Bellec at the University of Montreal, and he's doing connectivity analyses, and he's proposing a data-driven parcellization of the brain. So what we did is that we divided or parcellized the brain in 20 homogeneous regions in terms of the fluctuation of the bold signal. And here you have, that's very annoying. And here you have the 20 different regions that were parcellized within the brain. Then you are able to look, for instance, the first region is the superior cerebellum. Then you can look at how the first region is changing its connectivity with the other 19 regions, and within itself. You can look at this across different sleep stages, and if those changes across sleep stages are also different in young and older subjects. So you can imagine that this makes a lot of data to analyze, and I will not present to you the entire data set, but I will just give you a glimpse of the results that we have. So first of all, I just want to show you that if you look at the young subjects, this is showing the percentage of connection that showed significant changes in functional connectivity between different conscious states. So you can see in the young subjects that almost all the sleep stage comparisons showed very significant percentage of change in connection. For instance, if you take if you take non-REM 2 versus wake sleep, so it means that more than 50% of the brain is changing its connection between stage 2 and wake. In the older subjects, you can see that there are less comparisons between sleep stages that showed significant change, but remember that we didn't have enough stage 3 sleep to make some comparisons. So the message to take is that in older subjects, if you compare non-REM 2 sleep with non-REM 1 sleep or non-REM 2 sleep with wake, about 45% of the connection, of the brain connection are changing also as well in the older subject. And on this panel, you see the difference between older and young subjects, so meaning what is the percentage of connection that showed significantly age differences, so that were different, that the changes in connection were different in young and older subjects. And this was the case only for one stage comparison, is that if you compare non-REM 2 sleep with non-REM 1 sleep, and that was the case for about 16% of the connection of the brain. So this is showing the discovery maps for non-REM 2 and non-REM 1, because now I will only focus on non-REM 2 and non-REM versus non-REM 1, since I'm talking today about age-related differences, and this is the only comparisons that did show age-related changes. You can see, so here you have all the different, the 20 different regions, and it's showing for each of those regions, for the young, the older, and the difference between the young and the older, the discovery, percentage of discovery, which means the percentage of change with the other network. You can see both in the young and the older subjects that some brain areas change 60% of their connections with the other brain areas. And here, because this is what is mostly interesting to us, if you look at age-related differences, you can see that some, and actually the structure that shows the most interaction with age were, the basal ganglia. But almost all network, actually almost all seed regions, showed some very significant age-related changes also with the amount of connection. So this is just to give you a glimpse again of the kind of results that we had. So this is for, and follow me, for 3 seed regions. So the inferior DLPFC, the superior DLPFC, and the basal ganglia. So you have the regions that is in white to express what is the region. If you see green, and this line is showing the young subjects, this line the older subjects. If you see green or blue, it means that there is a decrease in functional connectivity in stage 2 sleep versus stage 1 sleep. So you can see that the young show for these 3 regions a decrease in functional connectivity, which is exactly what we were expected since sleep is getting deeper from stage 2 in sleep. You can see here, this is expressing the age-related differences between the young and the older subjects. And if you see yellow, it means that there is a significant difference between the age group. A thing that you probably notice also is that if you look at the line of the older subjects, you see decreases, but you see also some yellow, orange, and red color, which means that functional connectivity increased between stage 2 sleep versus stage 1 sleep in the older subjects. And as I said, this is showing the significant differences. So there are 2 kinds of significant differences that we can see. There is some of the age-related difference that we were expecting. For instance, the superior DLPFC is decreasing its functional connectivity with the superior temporal lobe, but this is not the case in the older subjects, and this leads to a significant difference. And there were some age-related differences that are more difficult, less expected, meaning, for instance, if you look at the basal ganglia, it increases its functional connectivity with the posterior motor central and with the insula very strongly in the older subjects, but this effect is not at all occurring in the young subjects. So in conclusions, age-related differences in functional connectivity, there are some age-related differences in functional connectivity in non-REM 2 versus non-REM 1 in about 16% of the brain connections. Compared to the young, older subjects show shallower decrease of functional connectivity or increase in functional connectivity. And those age-related differences particularly engage basal ganglia and frontal median temporal and lateral occipital network. And now we are trying to see how those changes can be explained with the changes in sleep that we were recording, and also if they are linked to cognitive abilities and brain plasticity. And I just want to finish with a little wink to REM sleep, because I have been talking to you only about non-REM sleep since the beginning of my talk. REM sleep is also very interesting because you know that REM sleep sustained activity is mainly cholinergic, and that it differs from wakefulness because it has during REM sleep, you have very little input from other neurotransmitters like noradrenaline, serotonin, or dopamine. So it could be an interesting marker also in order to see how the cholinergic system is going well or not in dementia, for instance. So since new studies shown that cholinergic degeneration may be especially key to early cognitive impairment in Parkinson's disease, though other neurotransmitters are also involved, we decided to see also, you remember I showed you that spindles were able to predict the development of dementia in Parkinson's disease. We decided recently to also go and look at REM sleep, actually, and we used spectral analysis. And this is a busy slide, but the take-home message, so we took spectral analysis to quantify the slowing of the REM sleep in Parkinson's disease patients. This is exactly the same cohort that I presented to you before. So 18 of the Parkinson's disease patients did develop dementia at follow-up. And we compared their slowing 4.5 years earlier. And actually, look at this slide, for instance, this panel. You can see that in red, you have the Parkinson's disease patient that did develop dementia 4.5 years later. And already they had a very strong slowing of their REM sleep also. So there is the reduction in spindle, but also the fact that the EEG, waking EEG, not waking EEG, but the REM EEG is also slowing. And this effect, if you look, it's different from the normal control, but also from the Parkinson's disease that did not develop dementia. So I just want to finish by thanking my precious collaborators, my students, and all my researchers that are working with me. Thank you.